Alright, here are the 5 reactions of the pyruvate dehydrogenase multi-enzyme complex. Here's a diagram from your textbook again. We've already seen this, and we already also saw the animated explanation of it just previously. And these reactions are very important. They actually are potential short answer material for exam 4, so please make sure you learn them. They're very important. And just to show you, here's a picture of this part right here. In E3 dihydrolipoyl dehydrogenase, there's an FAD group involved. Here's actually showing you that FAD group and how here's the disulfide right here. There's your disulfide, and there's your NADH, and how electrons are transferred between FAD and NAD plus to give you NADH. Just to show you a picture of that part of the active site of E3. Here's actually text explaining what I've just explained when I went over the animation and the reaction, but just if you want to look at that, here's some text for you explaining that. This is also a very important slide. The coenzymes and listing the coenzymes and prosthetic groups of pyruvate dehydrogenase. Okay, there are actually 5 coenzymes, like I've mentioned, involved, and here are what they are, where they're found, their location, and their function. The 5 coenzymes involved are cofactors involved. The first one is thiamine pyrophosphate, or TPP. This is bound to E1, and what's its function? It decarboxylates pyruvate, as we've seen. Lipoic acid is the second cofactor. Where's its location? We've seen this. It's covalently linked to a lysine on E2 and forms the lipoamide arm. We really know there's 2 of them on E2, where the acetyl group is transferred between them, and this accepts the hydroxyethyl carbanion from TPP, and you get acetyl lipoamide. Finally, or not finally, the third coenzyme is coenzyme A. It is actually a substrate for E2. So this coenzyme A is a stoichiometric cofactor in that it is converted to something, right? It is converted to acetyl-CoA, so it's used up in the reaction and converted from coenzyme A to acetyl-CoA, and it accepts the acetyl group from lipoamide, so it's a stoichiometric cofactor because it's converted in the reaction, whereas these other cofactors, TPP and lipoic acid, are catalytic cofactors that regenerated after the reaction. Here's FAD, the 4th cofactor. We've seen where it's involved. It's bound to E3 and is involved in in converting the lipoamide back to its oxidized form, so the FAD is reduced by lipoamide. It's also a catalytic cofactor because it's regenerated after reaction. And finally, here's your 5th cofactor, or coenzyme NAD+, and NAD+ is a stoichiometric cofactor because it is converted to something else in the reaction. The NAD+ is converted to NADH, so it is also a stoichiometric cofactor. So coenzyme A and NAD+ are stoichiometric cofactors because they're converted to something by the reaction, whereas TPP, lipoic acid, and FAD are catalytic cofactors because they're regenerated after the reaction. NAD+ is the substrate for E3, and it's reduced by FADH2, and NAD+ gets converted by reduction to NADH, which of course is high energy electrons, which goes on to make ATP in the electron transport chain / oxidative phosphorylation. Just one more thing to point out, here's your lipoamide arm right here. It's actually lipoamide right here attached to a lysine. So there's your lipoic acid that makes the lipoamide, because you have an amide bond here to the lysine. So this is a long arm, and I've mentioned this before. We saw this in the animation. The lipoamide arms work like a crane, shuttling the acetyl group between one part of E2 to another part of E2. Just to show you how this looks like, and this looks like in chemical structure compared to in the animation. And actually, here's what it looks like in the active site of the enzyme. Here's your lipolysyl arm that's involved in transferring the acetyl group between one part of E2 to the another part of E2, and then also, of course, to the coenzyme A to give you acetyl-CoA. And we've seen that in the animation and in the reaction that we went. One more thing to point out as well, we've talked about how arsenate uncouples phosphate formation. Right? We've seen this already when arsenate poisoned the GAP3 dehydrogenase reaction. What did we talk about? How arsenic is very similar chemically to phosphorus, so how it substitutes for phosphorus in biochemical reactions and is thus poisonous. Thus arsenic in this case disrupts ATP production through several mechanisms. Here's one of them that we've seen previously when we were looking at glycolysis and how arsenate poisons the GAP3 dehydrogenase reaction. Now, we've also talked about how arsenic is a heavy metal, and heavy metals have a high affinity for thiol groups. Arsenic, thus, in its arsenite form, in this form right here, has a great affinity to bind thiol groups, like these two thiol groups that are found in the lipoamide arms of E2. So arsenic, in its arsenite form, binds to the lipoamide arms in its reduced form, the thiol form of the lipoamide arm in E2, and makes a covalent bond to that arm. So what's this doing? This is irreversible inhibition. You have a covalent bond between the arsenic and the thiol form of the lipoamide arm, and thus the enzyme is poisoned and can't work when this happens. So arsenic is a poison to the E2 subunit of the PDH multi-enzyme complex as well. It irreversibly inhibits it, and thus is it's a metabolic poison through this mechanism as well. We've mentioned it's a metabolic poison through several mechanisms. Here's one way it's a metabolic poison as well. Okay, now let's start looking at the citric acid cycle itself. We've seen how pyruvate is converted to acetyl-CoA by pyruvate dehydrogenase, or PDH for short. Now let's look at the first step of the TCA itself, and this is catalyzed by the enzyme citrate synthase. Okay, what happens in this reaction? Acetyl-CoA and the energy of its high energy thioester bond is used to transfer the acetyl group from acetyl-CoA to oxaloacetate, a 4-carbon molecule, a 4-carbon carboxylic acid, to give you citrate, a 6-carbon carboxylic acid, and we go from a dicarboxylic acid to a tricarboxylic acid in citrate. So let's look at this reaction in more detail. Here is oxaloacetate, here is acetyl-CoA, and here are two important residues in the active site of citrate synthase. There's an aspartate, which works as a general base, and it deprotonates that H, which is actually the alpha carbon that's adjacent to a carbonyl of acetyl-CoA. And if you remember your OChem 2 well, you'd remember that if you have a carbonyl and you have an alpha carbon next to a carbonyl, the H on that is more acidic because of resonance stabilization. Once you lose that H by the carbanion, you actually get an enolate, right? If you remember this from OChem 2. So this aspartate can deprotonate, can pull off this H, which is more acidic than a normal CH. You get a carbanion, and you can look at it as a C- right here, or if you push electrons here and here, you can look at it as an enolate right here. Next, the enolate right here attacks the carbonyl, and this is like a mixed Claisen condensation. What is that? An ester. Okay, but here's your ester, but it's not an ester, it's really a thioester in acetyl-CoA. You've made an enolate of that ester. So there's your alpha carbon. You made the enolate by deprotonating the alpha carbon, and that enolate now attacks the carbonyl of a ketone. So that's why I said it's like a mixed Claisen condensation. So your enolate attacks the carbonyl of a ketone. This is stuff you've learned in organic chemistry too, and you get a bond between this carbon here, because that's the carbon that made the carbanion by deprotonation, between that carbon and the carbon of the carbonyl of oxaloacetate. So we get that, and you have this bond shown here, and the colors help you see what came from what. There's your acetyl group, there's your methyl carbon of acetyl group, it's lost an H, and now has a bond to the carbon that had the carbonyl of oxaloacetate that now has been protonated and has an OH on as well. So this carbon goes from an sp2 carbon to a tetrahedral carbon right here. So this is an example of actually classic coenzyme A chemistry, where coenzyme A likes and loves to do these kind of mixed Claisen condensations, where you make a carbanion. That carbanion, of course, is part of an enolate on the thioester as shown here. The carbanion attacks, or the enolate attacks, a carbonyl, and you can get condensations like this. Coenzyme A likes doing condensations like this. The last step then is you've got to just hydrolyze this high energy thioester bond right here. What's hydrolysis? You just add water to it, you end up with a carboxylic acid, and that gives you citrate right here, and you end up with coenzyme A released as well. And of course this H right here, that's the important thing, and the energy, from this the hydrolysis of this high energy thioester bond is what drives the reaction. This, as you can see, is a one directional arrow, so that's what drives the reaction, because these two steps right here are reversible steps. Alright, so here's the reaction catalyzed by citrate synthase. Here's acetyl-CoA, there's oxaloacetate. That methyl group on acetyl-CoA loses a carbon, loses a hydrogen, excuse me, you get a C minus. That C minus attacks this carbonyl carbon of oxaloacetate. There's a bond formed between this carbon here and that carbon. This O goes to OH, and you get the acetyl group added on right there. It's in red, so you can see how it's added on, and you get citrate, a tricarboxylic acid, because it has 3 carboxylic acid groups on it, excuse me. If you look at citrate synthase, you'll see it has this pinwheel-like structure, and as it binds substrates, it closes in on the substrates, right? So what does that mean? If you see an enzyme closing in on its substrates, it's an example, of course, of induced fit. You can see this in this diagram as well. Here's the open form. When it binds its substrates, for example, oxaloacetate, it closes in on its substrate, and you get induced fit exhibited here.